Hello, everyone, and welcome to today's webinar, Advanced Solutions for CRISPR Gene Editing. This webinar is part of the CRISPR Virtual Event Series 2024, brought to you by LabRoots, and is sponsored by GenScript. For more information on our sponsor, click the GenScript logo found on your screen, or visit genscript.com. I am Israel Gonzalez of LabRoots, and I will be your moderator for this webinar. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by both communicating with other attendees using the group chat feature and also by submitting questions during the presentation. I'd like to now introduce our speaker, Rumen Bugoev, Head of Segment Management at GenScript USA. You can view our speaker's complete biography below. Rumen, you may now begin your presentation.
Thank you, Israel, for the kind introduction. It is my pleasure today to introduce our audience to the advanced solutions for CRISPR gene editing by GenScript. This is the agenda for today. First, a little bit about GenScript, then introducing gene editing applications, GenScript CRISPR solutions, and why you may want to choose GenScript as your provider. So first, a little bit about GenScript. GenScript is a global company. We have over 200,000 customers. We operate in over 100 countries. We have multiple locations, production locations. We have 2 production sites in US, one in the East Coast, one on the West Coast. We have multiple production sites in China and production site in Singapore. We have many commercial offices around the world. We operate in over 100 countries. We have over 6,000 employees, more than 800 R&D staff. We have many patents and patent applications, and we have supported our customers in publishing over 100,000 peer-reviewed publications using our services and products. So if we look a little bit more at the GenScript group, consists of several entities. Today, I'll introduce you only to 2 of these entities on the left side, the Life Science Group and the GenScript ProBio CDMO division. But very briefly to introduce the other 2 entities that we have. Bestzyme is our synthetic biology division. They focus on human and animal food supply engineering, and they have very large capacity. Legend Biotech is an innovative cell therapy company. They launched their first CAR-T cell therapy drug, Carvykti, for multiple myeloma, working with Janssen. And they have advanced pipeline making progress in therapeutics for leukemia, solid tumors, infectious disease, and other indications. And again, my talk today is going to be on our capabilities for the GenScript Life Science Group and the CDMO division. The Life Science Group contains our CRO division, also our product division. We use a lot of automation in our CRO operations to increase our efficiency. Our product division also offers a lot of products for automating your process instrumentation and other innovative products and services. Our biologics CDMO division has very large GMP capacity. They have very strong regulatory experience. They have been supporting a number of customers in their preclinical and clinical work. And they're one of the leading biologics CDMO service providers. So now a little bit about the gene editing applications. Of course, gene editing has many different applications in many areas of the research, but some of the more common ones are for cell therapy and gene therapy. Cell therapy is very often, the gene-edited cell therapies are very often referred to as ex vivo gene editing. Basically, for cell therapy, cells are typically extracted from the patient or from another donor. Then they're engineered externally, either using viral or non-viral methodologies for gene editing. then it may be grown further and then formulated and introduced back into the patient for treatment. Well, for in vivo, for gene therapy, usually the whole engineering is done inside the patient body using either viral or non-viral methodologies. And if we look at the current pipelines for therapeutics, for gene, cell, and RNA therapeutics, we see that there are over 4,000 therapies in development. And about half of them, a little bit more than half of them, are actually using some kind of gene editing for the therapeutics. They're developing either cell therapy or gene therapy. If we look a little bit more into the pipeline, we see that many of those are preclinical. Over 1,400 of them are in preclinical stage. Maybe about 600 of those are in various stages of the clinical trials, phase 1, 2, and 3, and 5 pre-registration. So a lot of activities in the therapeutic area using gene editing. So now we have two general methodologies for gene editing, viral gene editing and non-viral gene editing. For viral gene editing has been the workhorse of gene therapy industry for a while now. Viruses are very efficient at delivering their payloads into the cells and very efficient at expression. Non-viral gene editing has maybe some more challenges with the delivery of the payload. However, is considered typically safer, also offers a lot of flexibility and a lot of options. I'm going to go over this a little bit later. So if we look at the safety profiles, viral vectors have more concerns about immunogenicity, potential immune response, cancerogenicity, potential long-term effects from using virus for gene editing. Well, the non-viral approaches don't have that many concerns for the safety. However, there are still some challenges about the efficiency of the gene editing, also about the specificity of the gene editing with the non-viral approaches. And some of those are solved by some of the newer methodologies. If we look at packaging capacity, viral vectors are somewhat limited due to their genome size constraints, while the non-viral methodologies do not really have constraints in the payload size. However, of course, the bigger the payload, the higher molecular weight, the harder it is to transfect. So those are some challenges also in the non-viral payload with very large molecules delivering. If we look at the non-viral gene editing methodologies, there are a number of those on the market. Zinc finger proteins will be some of the original methodologies being used to edit DNA. They rely on DNA-protein interaction. Binding site is somewhere between 9 to 18 base pairs, and usually performs double-stranded cleavage, performed by FOC1 endonuclease. And in order to do the editing, these zinc finger proteins, their domains, they're assembled to bind to the specific sequences and then linked to FOC1 endonuclease. And then the two protein complexes, one for each DNA strand, are being linked with a bridge. One of the challenges of creating these zinc finger proteins and complexes for cleavage is that actually zinc finger domains can influence each other. So depending on a neighboring zinc finger domain, the other could be influenced in their specificity. So that makes it a little bit more challenging. Also, in order to perform the gene editing with zinc finger methodology, you have to design novel, basically protein complexes for each new sequence you would like to cleave. And so that's a significant roadblock in gene editing. Another technology for gene editing is TALEN. TALEN has been very well investigated. There are the tail domains that bind to specific DNA sequences. Those are very well characterized and they're relatively easy to assemble into a sequence to bind to specific DNA sequence. Also relies on DNA-protein interaction. And also uses the FOC1 endonuclease for the cleavage. However, the two protein complexes, the tail with the FOC1 for each DNA strand, strand, they don't need to be linked with a bridge. So that makes it a little bit easier to engineer. Also, the well-characterized domains make it relatively easier to engineer. But again, you do have to engineer different sequence of tail domains for every DNA sequence you would like to cleave. So CRISPR-Cas system actually introduces a lot of flexibility and ease of use into the gene editing, because it's using relatively short RNA sequence for targeting. So the enzymes are usually the same enzyme for every DNA sequence. And then the only thing that needs to change is the guide RNA that is being used to target the specific DNA sequence. So that provides a lot of flexibility and ease of use into the gene editing. There are also a lot of tools that are being created, a lot of different Cas enzymes that can be used for the cleavage. So if we look at a little bit more in the CRISPR-Cas system, there are different enzymes that can be used that provide a lot of flexibility. Cas9 is one of the first enzymes that was introduced for gene editing. It does one double break cleavage, cleaves 3 bases away from the binding sequence, and after cleavage, the enzyme is released. The Cas9 usually prefers G-rich PAM sequences, and it's pretty efficient at the cleavage and has a lot of flexibility in the editing, because it can introduce base modifications, insertions, deletions, and so on, and you can insert relatively large fragments also. Another enzyme that is also finding application in gene editing, another Cas enzyme, is the Cas12a for DNA editing. It introduces staggered breaks that has some advantages for insertion of genes. It cleaves about 18 to 23 bases downstream of the PAP sequence, and the enzyme stays bound after cleavage, which allows it to cleave other single DNA strands that are close by. It prefers T-rich PAMs, so it reduces some more flexibility of how we can do gene editing depending on the application. One enzyme could be used or another, preferred depending on the purpose and the sequence that is being edited. Cas13a allows us to edit RNA, so it cleaves single-stranded RNA, cleaves downstream of the binding sequence, and also stays bound to the RNA after cleavage, so it can cleave other single-stranded RNAs that are close by. So these two enzymes, Cas12a and Cas13a, because they stay bound to the original nucleic acid and they can cleave other neighboring amino acid DNA sequences, they have found application also in diagnostics. So some newer CRISPR modalities have been introduced in more later years to address some of the challenges with the original CRISPR-Cas systems, because the original CRISPR-Cas systems have some issues with non-specific alterations, or so-called indels, insertions or deletions that are non-specific. And these new modifications, the base editing, uses deamidase, cytosine deamidase and adenosine deamidase that are linked to nickase enzyme correspondingly, and they can convert basically C and G to TNA and A and T to G and C. So they're very efficient at base conversion. Also, they have fewer non-specific modifications than the original Cas9 enzymes. However, they're only capable of single base modification. In not all disease conditions, only 50% of the disease conditions are due to single base modification. So they have somewhat limited applicability because of this limitation. Prime editing was introduced later to address some of these limitations. Prime editing is done by having nickase linked with reverse transcriptase, and the nickase performs a single cleavage, and then the reverse transcriptase uses the guide RNA, the PEG RNA, to synthesize the modified chain of DNA. And then the DNA repair mechanism replaces the original, usually with a new synthesized DNA chain. So that has the advantage that actually multiple base modifications can be done at the same time, insertions, deletions, modifications. However, this methodology is also limited to a relatively small number of bases. Around 50 or so bases can be modified compared to the base editing, which works only on single base. However, the Cas9 and Cas12 methodologies, enzymes can have also larger inserts, which are very beneficial in some situations. So looking more in the different CRISPR modality, Cas9 is very flexible, can do wide range of edits, deletions, insertion, modifications, can insert very large fragments. However, it's not perfectly specific. It has some, it does some non-specific modifications. Base editing and prime editing are more specific than Cas9. However, base editing is limited to single base modifications, and prime editing is limited to small number of modifications. About 50 or so bases can be affected. Additional challenge with base editing and prime editing is the enzyme complexes that are now created to perform the editing, they're relatively large. So in some situations when it's desirable to use viral vector for transfection and for delivery, this is becoming more challenging for base editing and prime editing because, for example, AAV viruses their genome is too small for accommodating these large protein complexes. So what are the CRISPR solutions by GenScript? We have complete solutions from beginning to end. We can support discovery work, lead generation, preclinical, clinical developments. In discovery work, we have a lot of different CRISPR libraries for knockouts, for transcriptional activation. We can develop custom libraries for target validation. For the lead generation optimization, we have number of reagents. We can synthesize sgRNAs, crRNAs, PEG RNAs, depending on the enzyme being used. We have number of enzymes. We offer off-the-shelf Cas9, Cas12, Cas13, other variations of them. We can synthesize HDR templates. For preclinical work, we can increase the quality of these materials to GMP-like or ISO 13485. And then for clinical work, we can offer this as cGMP quality services and products. So basically, we can support development from beginning to end, offering variety of different services and products with phase-specific quality systems. And this is little selection guide, what different enzymes we have and what they're recommended for. For example, for very efficient knockout or knock-in, SP-Cas9 does very good job for that. However, if higher specificity is required, fewer non-target edits, then ultra ESP-Cas9 may be preferable to use. We also have SP-Cas9 linked with GFP for various fluorescent assays, flow cytometry, immunofluorescence, and so on assays that can be done using this modified SP-Cas9. We also have the Cas12, several versions of Cas12. They offer a lower molecular weight, so they're easier to transfect using viral vectors. Also, we offer SA-Cas9, which also has lower molecular weight for convenient AAV transfection. And we offer two versions of RNA cleavage enzymes, Cas13A. We offer different type of DNA payloads depending on the purpose. They have different advantages and disadvantages. We offer gene-exact single-stranded DNA HDR template, usually suitable for T-cell therapies and animal models. Offer minimum cytotoxicity, very precise knock-in. minimized off-targeting effects. However, the limitation is that the fragment that can be inserted is more than 5 kilobases. We offer this single-stranded DNA as research in GMP grade. GenWant double-stranded DNA is another HDR template that we can offer. It's usually good for scale-up in larger quantities and also larger genes. Up to 10 kilobases genes can be inserted using this template. It uses closed ends. The double-stranded DNA ends are closed for higher stability and higher accuracy, and more suitable for scale-up application. As I mentioned, up to 10 kilobases fragments can be inserted using this template, and we offer it in research in GMP grade. And then our GenCircle double-stranded DNA template. It's very good it's essentially miniaturized vector where we have removed a lot of the unnecessary genes. So we have minimized the size to 429 base pairs only. And it offers very high efficiency for knock-ins, about 30% higher efficiency than other templates, and is very efficient. And we can offer it in research grade and in GMP grade. Up to 50 kilobases fragments can be inserted using this template. Here are just some examples showing the GenCircle double-stranded DNA being used compared to a plasmid. It's usually a better replacement for plasmid templates. So in this case, you can see that the efficiency of GenCircle is much better than the plasmid template. Another case study used as transposon vector for knock-in, 4.8 kilobases insertion. You can see also the performance of the GenCircle is much better than the performance of the PAC plasmid that was used as a control. We have also excellent software for HGRNA and HDR templates design. It's very efficient. It does very good prediction. It has very good prediction capability. Predicted, as you can see, our HGRNAs compared to the competitors here are with better predicted efficiency, better specificity, and lower off-target modifications. On the lower left chart, if you look at there, you can see that our HGRNAs perform better than the ones designed by the competitors. And here in the middle chart, you can see different genes being edited with HGRNAs developed using this software, and it's very efficient. In addition to custom synthesis, we also offer a lot of HGRNAs that have been developed by Feng Zhang's laboratory at the Broad Institute under license. We license from them. And we have full genome coverage. Basically, we have full genome coverage for human genome, for mouse genome. All the genes that are in the genome, we have usually multiple HGRNAs that are targeted to each gene for knockout, and multiple HGRNAs that are targeted for transcription activation for each gene. So those are also available for purchasing. We also have very extensive CRISPR cell line services. If you're trying to do CRISPR cell line knock-in or knockout, we offer a number of services for knock-in, knockout, also whole gene deletion, and so on. mRNA is being used as a delivery methodology very often. It has some advantages because of its transient nature. So it's preferred for safety reasons, it's preferred compared to other methodologies because after some time the mRNA is degraded and basically no longer doing any off-target edits to the organism. We have full service from beginning to end, starting with codon optimization. We have ready-to-use optimized UTR designs that we can offer you for use. We also can do the codon optimization with over 200 different factors for the optimization. We can do the gene synthesis as a single fragment or multiple fragments. We can perform the cloning and plasmid preparation, also plasmid linearization for the IVT reaction. We can prepare any custom enzyme if required. We have a lot of experience developing various enzymes. And then we can synthesize the mRNA in different formats, a linear format or circular format. We can also package the RNA into lipid nanoparticles. And we can optimize these particles for delivering specific cell types or specific tissues. This is just comparison of different IVT RNA payload formats. We offer linear mRNA, we offer self-amplifying RNA and circular RNA. They have different advantages and disadvantages. The best feature of the linear mRNA is that it has very low immunogenicity. Also, is very well investigated, has been used for a very long time. Some of the newer methodologies, circular RNA and self-amplifying RNA, they have stronger ability to for stronger expression. Self-amplifying RNA having the strongest than the circular RNA. They're also more stable than the linear mRNA. They have a little bit higher immunogenicity. However, the payload, the amount of RNA you need to inject may need to be lower, maybe lower compared to what you may need for regular mRNA, linear mRNA. And this is just an example of EGFP RNA transfected into HEC293 cells. You can see that the linear mRNA starts, the signal intensity starts declining at 72 hours compared to the 48 hours, while the circular RNA is still going strong and increasing even in the 72 hours compared to 48 hours. In the self-amplifying RNA, delivering the strongest signal and even slightly stronger at 72 hours compared to the 48 hours. Again, because of its higher stability, and also the self-amplifying, of course, amplifies itself and produces stronger signal. We also guarantee our poly-A tails. Poly-A tails are, of course, very important for the RNA stability and expressibility. We have special methodologies for producing the poly-A tails with the desired length. Anything up to about 100 adenosines can be added, and we can produce the RNA with high consistency and high uniformity. Here is some customer feedback for our mRNA expressions. A customer at Georgia State University using EGFP expression in mammalian cells. Then another customer using mCherry expression in primary cells in Upstate Medical University. Another customer using our mRNA, FLUC mRNA for mouse models with great success. And then we have some positive responses from Merck using our mRNA as antigen and XAP using our custom mRNA for expression. We also have some catalog mRNA. If you'd like to try mRNA format for your gene editing, we have ESP-Cas9 mRNA off the shelf that you can order and be delivered in a couple of days. ESP-Cas9 mRNA and ETRAC sgRNA and LNP particle. We have ESP-Cas9 circular RNA, CPF1 mRNA, and we have some of the prime editors, prime editor 2 and prime editor 3 as mRNA. So we also have other mRNAs for gene editing and proteins and enzymes. Feel free to look at our website and select something that may be of interest. So mRNAs are usually delivered in using different methodologies. Lipid nanoparticles is the most popular. Adeno-associated viruses sometimes used. Sometimes peptides are used for delivery, cell-penetrating peptides. liposomal transfection, exosomal transfection, and so on. And we can support all of these transfection systems with our capabilities. So if you look at viral vector delivery, because viral vectors are very efficient at transfection, we can see that, as I mentioned before, some of the Cas enzymes, they can fit inside an AAV vector, which is a lot of the time the preferred vector for delivery for gene therapy, for example. SP-Cas9, SA-Cas9, CPF1, those are enzymes that can fit inside AAV vector for delivery. However, some of the newer modalities that are the base editors and the prime editors, they're a little bit too big for the AAV vector. So for them, adenovirus vector or lentivirus vector may need to be used. Or some different methodology may have to be created. Some papers have publications showing that the editor may be split in two parts and then transfected separately and so on, but different design strategy may need to be deployed in order to use AAV vector for those for the base editors and prime editors. We have also full AAV engineering capabilities. We can start with codon optimization. We can perform the gene synthesis, or we have very large ORF collection that you can use. Then we can do further engineering, genome engineering, in order to provide target-specific promoters or enhancers, maybe some regulatory elements like miRare, and so on. We also have AAV ITR guarantee. We have specific strains designed to make the ITR regions more stable, and we have proprietary technology for sequencing these GC heavy ITR regions to show the sequence is intact. We can also support capsid engineering to enable specific tissue tropism. We can perform either mutagenesis services, or we can supply you with mutant library for your engineering of the AAV virus. We have also full service lentiviral capabilities, starting from gene synthesis, of course, codon optimization, gene synthesis, subcloning, plasmid preparations, lentivirus packaging, quality controls, multiple methodologies for ELISA, flow cytometry, mycoplasma, Sanger sequencing, and so on. And our QC, of course, is always customizable to your requirements. We can also prepare the plasmids, the viral plasmids, or the lentivirus or AAV virus under GMP conditions for your clinical trials or GMP manufacturing. We also have some other accessories, like we have a lot of Cas enzyme antibodies that can be used for various detection techniques. And we have been supporting CRISPR genomic medicines for the past several years. We have a lot of expertise also in GMP production. We have done over 100 GMP batches. We have passed successfully over 22 audits in different regions in the world. We've supported 30 IND projects, and we have supported 6 successful IND applications for our customers. This is just a case study where we supported human clinical trial. For this customer, we designed for them two-component sgRNA, and basically they were working on very innovative CAR-T therapy using integrated PD-1. And they were doing a human clinical trial 1 with 8 different patients, and 5 of the patients achieved complete remission. And these are some of the key services and product offerings that we have. Basically, a lot of these services and products, we can offer them from R&D, from research grade all the way through GMP, antibody and protein expression, CRISPR, gRNA and HDR templates, plasmid preparation and purification. For IVT mRNA, we support research and preclinical grade at the moment. Our GMP is coming soon. AAV vector, we can support from research grade all the way to GMP grade, lentiviral vectors, from research to GMP, peptide synthesis from research to GMP. We have some products also here that we offer. The cell isolation system is a very novel instrument for cell isolation, can purify T cells, NK cells, stem cells, and other cells using nanomagnetic beads with antibodies. We offer this system for cell therapies. We offer this system in research grade, and we also for GMP grade. We also have excellent T cell activation reagent that we offer for research grade and GMP grade. And we have some other automation. We have automated plasmid purification system and automated protein purification systems using magnetic beads that can be very efficient for research applications. So why using GenScript? GenScript is #1 provider in gene synthesis in the world. We have synthesized more than 30% of all synthetic genes worldwide. We have very strong experience in gene editing, mutagenesis and cloning, gene libraries. We have supported many customers, over 200,000 customers. We have synthesized over 3 million genes, and we have very diverse experience in all areas in CRISPR and genomic research and proteomics research. We also supported our customers in publishing a lot of publications. We have over 100,000 citations at this moment, and many of publications went to very prestigious journals such as Cell, Nature, PNA, and so on. And these are just some pictures from our GMP facilities. We have 2 GMP facilities in China, one in Nanjing and one in Zhejiang. And we have GMP site in US, in New Jersey, Hopewell. We recently opened GMP site also in US. And this is my presentation for today, and I'll be happy to answer any questions you may have. Thank you for your attention.
Thank you very much, Rumen, for that informative presentation. Let's go ahead and dive into the Q&A segment of the webinar now. And as we do, I'd like to continue to encourage our audience to submit your questions during this time. Our first question is, can you produce custom Cas enzyme to our specification?
Yes, so we have very strong experience. We have developed literally hundreds of different Cas enzymes for different customers, for internal use and for external use, for external customers. Cas9, Cas12a, Cas13a, MAT7, different base editors and prime editors. So we have very strong capabilities in that regard. We can develop those enzymes and deliver them in 2 to 4 weeks. We guarantee the purity and yield. We can provide any basically Cas variant. And we have very flexible scale that can start from 500 micrograms and go to very high quantities. In addition, we can offer add-on service. We can use our AI-powered enzyme design for functional improvement. And we have various assay formats that we can perform for you.
Great, thank you for that. Our next question is, what sgRNA libraries do you offer?
So we offer a variety of sgRNA libraries. We can design a lot of custom libraries, knockout libraries for a group of genes, specific pathways, genome-wide knockout libraries. We also offer the Broad Institute knockout or transcriptional activation libraries that we license from them. And those have been already pre-tested and shown to work.
Perfect. It looks like we have time for just one more question here. And let's go to end with this one. Can you produce the custom Cas enzymes in GMP grade, and what is the timeline for something like that?
Yes, we can definitely produce the Cas enzymes in GMP grade. Usually the timeline for this is somewhere between 6 to 9 months. We can confirm the batch-to-batch consistency. We can do the process development. Again, we can also offer some improvements of your design. If you require, we can use our AI platform to improve the functional performance of the enzyme, additionally to just the production.
I see. Thank you so much for that. And thank you again, Rumen, for your time today and your important research. Any questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you registered with. This webcast can be viewed on demand for 1 year until October 23rd, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Thank you to our audience for joining us today, and again to GenScript for sponsoring this webinar. Until next time, goodbye, everybody.
Thank you for your attention. Goodbye.